Hello everyone and welcome to this webinar. My name is Nuria Roldan and I work as a project leader at AlveoliX, an organ-on-a-chip company that has been working in the last years in establishing an organ-on-a-chip model of the alveolus. Today I will be talking about how systems like this, like the lung-on-chip, are boosting the introduction of the 3R concept into research, drug development and toxicology from the lung perspective. But first, which systems do we have available to study the lung? For sure, the first-hand information from the patients at the clinics is the most reliable and close to applications for humans, followed by the research that we can do in their tissue samples and also in ex vivo systems. But if we think about it, as it comes from the patients, this information is rather limited, and besides, healthy tissues or individuals are often missing to compare. If we want to get knowledge about the biology in health and disease, we need a model allowing to turn on and off the pathways to understand how they function, or also introduce different disease triggers. So this, of course, is something we cannot do in humans, and that's why animal models have been used for so long during the years. Even though using animals have provided a lot of valuable information, more and more evidence is pointing that these models do not replicate human physiology as well as we thought they did. And besides, for obvious reasons, they raise multiple ethical concerns. So if they're not predictive, which alternatives do we have? Yeah, sure, we have in vitro systems that allow for high throughput, introducing modifications. They are normally simple to use, and we can also control the environment. However, they are too simple to extrapolate results to what happens in the human body. So, could we make these systems more complex and introduce physiological parameters to make them more predictive? The answer is yes, and this is how microphysiological systems were born. These in vitro models introduce different features of the in vivo situation, including mechanical forces, the 3D environment, or different cell types. They are what we call advanced in vitro models, and among them we can find, besides organs on chip, also microtissues and even organoids. This technology is proving its potential in many areas, including drug testing, and they represent a very good alternative to reduce and replace animal models. MPS can be applied at both basic and translational research, including the development of healthy and disease models that we can then use for toxicology, safety and efficacy testing, and also for personalized medicine. Putting this into perspective, here we have the example of the drug development process. Well, as you probably know, developing a new drug starts by screening thousands of compounds that are subsequently reduced to a few through in vitro, animal testing, and finally, clinical trials. Bringing a new drug into the market is a very long and expensive process, and MPS can help making it shorter and also more cost-effective. MPS can be applied at the preclinical testing and normally during the lead molecule optimization or during the definition of the candidate drug compounds, since they are more predictive compared to conventional in vitro systems. But more importantly, they are very good assets for safety and efficacy testing, and there is where the predictive potential can help accelerate the process and avoid, in some cases, unnecessary animal testing. Now that we have this overview, so MPS seem like a very good option, but they are quite complex, and this can sometimes be very challenging. So from the engineering perspective, which support, architecture, and forces we want to integrate is key to develop the right environment for the cells. In the case of the lung, for instance, we need to introduce a soft and porous membrane support to allow for cell culture. And ideally, this support should separate an apical and basolateral compartment that we can assign to an air and a blood compartment. Then, we need to introduce the forces simulating the breathing motion or also the blood flow. And last, if we think of the structure, an open design would allow for an easy access to the cells and to develop the chip for exposure applications. When it comes to the biology, first we need to think of which cell types we want to use. So we need to use stem cells, we want to go for primary cells, one cell type or many. This will then define which cell culture medium is better suited if we have a co-culture, and this will in turn modulate how cell-to-cell communication is established. Then in addition, we need to define the right microenvironment in terms of the ECM or cellular matrix, so we can make the cells to feel and behave as they would be at home. So you can imagine, with this many components, the options are almost limitless. In the very recent history of the lung on chip, different models have been developed for different parts of the lung, including different cell types and chip designs. So we have to be careful when we say lung on chip, because we have to consider that we may be modeling different regions of the lung that can be either upper airways, small airways, or also the alveolus. The alveolus is the smallest functioning unit of the lung, and that's why I will be focusing on air-blood barrier models, some of which I have shown here in blue. All these models have something in common, and is that they require a particular structure and cell type to simulate the very thin epithelial endothelial barrier that is in charge for the gas exchange. So here I show you the lung-on-chip system we have developed. So this system is comprised by 3 modules, including the docking station that holds the chip itself and connects it to the other 2 modules. Then we also have the exchanger that provides the support for the medium exchange, and finally the breather, which allows to control the cyclic stretch, simulating the breathing function. So if we focus now on the cell culture plate, as you can see here, it has exactly the same dimensions as a 96 well plate. So the AX12 comprises 2 chips, as you can see here, one to the right and one to the left. And each of these chips includes 6 individual cell culture units, accounting for a total of 12. Each of these units is composed by 3 wells, as you can see in the green box. From these 3 wells, the central chamber of the chip holds a very thin and porous membrane that serves as support for the cells, and the cells can grow on either side of it. This membrane is very thin, it's only 3 micrometers thick, and it's flexible. Through the pores, the cells can also communicate, and as you can see here, by seating alveolar epithelial cells on top and endothelial cells at the bottom, we can recreate an in vitro system for the air-blood barrier that is very, very thin and holds no more than 15 micrometer thickness. And finally, we have the breathing function. This motion is inspired by the movement of the diaphragm, and in this case, what it did was introducing a microdiaphragm within each cell culture chamber. So this is a very thin membrane placed at the bottom of the cell chamber, as shown here in gray, that can be deflected by the action of the breather. So then, when the chip is filled with medium, this deflection can be transmitted to the membrane that holds the cells, shown in blue, providing a breathing-like stretch. The motion is in 3D, and then the settings are adjusted so it can simulate the physiological breathing pace in humans. In the video to the right, we can see the breathing function in action. As you can observe, the 3D deflection is clearly visible, with the cells in the outer part of the membrane being slightly less deflected than there was on the center, simulating then the in vivo situation. So with this model, in our lab we have developed different applications and also biomodels. So one of them is focused on inhalation. Through breathing, we are continuously exposing our lungs to particles of the environment. These particles can cause adverse reactions, like happens with pollutants or, for instance, with pollen. So we try to simulate a model that does exactly this. For this, we are developing a model together with VitroCell that allows combining the lung-on-a-chip technology with an aerosolizing chamber, opening up the opportunities to expose alveolar cells to different molecules of our choice and also observe their effects. For cellular models, here we have chosen to use human alveolar epithelial cells, which are the first cells that are encountered by any molecule that enters within the alveolus from the air side. As you see, these cells can form a very tight barrier that we can observe here in the graph to the right, represented by a very high transepithelial resistance or tear. Further applications of this model is to use it to study the effect of drugs that are inhaled, and they basically come through the inhalation route, or study new alternatives of drug delivery. Then in addition, this in vitro system could also serve to reduce or replace, in some instances, some of the animal experiments that are being used in inhalation toxicology. So here there are some examples of applications we have used the system for. First, we use zinc oxide nanoparticles, which are a very common constituent of many different products such as cosmetics, rubber, concrete paints, or even sunscreen. We use different concentrations of nanoparticles and we apply them in dynamic and static conditions on alveolar epithelial cells. We could observe that the breathing conditions made the cells more sensitive to zinc oxide-induced toxicity, particularly at the higher doses tested, which actually simulates an acute effect at a dose which would be in range of the maximum amount of zinc oxide nanoparticles accumulated for an exposed worker throughout their whole life. As a proof of concept, we have also used a molecule whose effects are very well characterized. And in this case, it was the well-known profibrotic inducer of TGF-beta. So we use this molecule in repetitive dosing using the cloud system. Here we can observe that exposure to this profibrotic cytokine induced the weakening in the barrier function, as we can observe here by the decrease in TEER. And this was also associated to a decrease in cell viability. The treatment with TGF-beta was also reflected in an increase in the mesenchymal markers, collagen 1 and alpha-smooth muscle actin, and a decrease in the gene expression on epithelial ones such as IC adhering. And this suggests that TGF-beta nebulization is inducing epithelial to mesenchymal transition in this model. In the following example, I show you a model we are developing for chip applications. In this case, we had a co-culture of alveolar epithelial and endothelial cells. Once the barrier was formed, we used a pro-inflammatory molecule in different doses. And we could observe that there was a dose-dependent effect on barrier function weakening, with a highest dose here, 4 times the dose, leading to almost a complete destruction of the barrier. This was also correlated with an increased permeability of the system to small and big molecules. Besides, we could also observe that endothelial cells were much more susceptible to this molecule than the epithelial ones, as we could observe both under the microscope and also by a much more pronounced cytotoxicity on the endothelial layer. Indeed, the lung-on-chip can also be used to study toxicology by following inflammation. Some lung inflammatory diseases, such as ARDS, can lead to pulmonary edema and immune cell infiltration, and this is a signature of an impaired barrier function and damage at both the epithelial and the endothelial side. So to model these conditions, we work normally in aquaculture models, such as the one I showed in the slide before, in which we use human alveolar epithelial cells and lung microvascular cells to simulate the capillaries. Then we treat this co-culture system with different molecules to simulate a disease, and then we can investigate new targets and also test the efficacy of novel drug candidates. We really believe that using systems like this, we can get very valuable information directly from human cells that can prevent or at least refine the conditions for certain animal studies. Okay, so keeping in mind the important role of immune cells during inflammation, we tried to model on the membrane if we could recreate the recruitment process. So for this, we seeded endothelial cells that you can see here in red, and we activated them with LPS. So it was very nice to see that PBNCs that you can see in green were being actively recruited by the activated endothelium. And after a certain period of time, we could observe that many more PBNCs were attached to the endothelial layer in the presence of LPS. But more importantly, these cells were not only recruited, they could also transmigrate upon attachment, going through the endothelial layer and then through the chip membrane towards the other compartment. Here there is another example of how important it is to introduce immune cells depending on the model. In this case, also triggered by LPS, we wanted to observe how we could develop a system trying to simulate the features for acute lung injury or ARDS induced by infection. So here we use LPS as the trigger, and we could observe that only in the presence of PBNCs we had actually a decrease in the barrier function. Here you can see that is quite a dramatic decrease in TEER. And this was also accompanied by an increase in the secretion of pro-inflammatory compounds, and in this case it was iolate. If you look what happens at the endothelial side, we can observe that only in the presence of PBNCs together with LPS there was a change in morphology, with endothelial cells losing their typical cobblestone shape towards a more spindle-like shape of the cells. And this highlights how important it is, again, the role of immune cells in modulating inflammation and toxicity, and why we need to include the immune component in many models when we want to recreate an in vivo situation. Another model we have been working on is on trying to simulate the features of idiopathic pulmonary fibrosis. So this disease is truly devastating, and it causes scarring of the lung tissue and ends up in fatal respiratory failure. So IPF is characterized by a thickening and stiffening of the interstitial spaces, in which normally an already sensitive epithelium, as this functional epithelium, starts undergoing an abnormal tissue remodeling, and then finally the alveolar structure is completely destroyed. So this disease has no cure up to date, and this is why predictive preclinical models for drug efficacy testing are essential, so we can accelerate the drug development process and bring a solution for these patients. For this, we have developed a model consisting on human alveolar epithelial cells that we co-culture with lung fibroblasts. And this can be treated with different profibrotic triggers, such as, for instance, TGF-beta, that is the one we are using currently. So this could be applied for target determination as well as for efficacy testing, as I mentioned before. And at the same time, it can serve as an alternative tool for different animal models that still do not resemble completely the human IPF features, as it happens with the bleomycin model. Here's some results of our experiments. So our data suggests that in our epithelial fibroblast co-culture, TGF-beta does not have a significant effect in decreasing barrier integrity. However, in breeding conditions, it seems that there is a trend of the treatment that it seems to decrease already the barrier function from 24-hour onwards. If we look at the toxicity induced by the treatment, we could detect that there was an increase in the epithelial cells at 48 hours from TGF-beta introduction. However, the fibroblast side was not affected by this treatment. Interestingly, the effect on the epithelium we could reverse by nintedanib, which is a drug that is currently on the market for fibrosis. The cytotoxicity levels were reduced to those of the untreated conditions. And these results support that the model could be a good preclinical tool to test the efficacy of new antifibrotic drugs. Finally, the lung-on-chip also permits to go one step closer to the clinics, as it is allowing to evaluate aspects that are reserved only to humans, such as patient-to-patient variability, or also applications such as those involving the human leukocyte antigen. So in the first place, to study this donor-to-donor or patient-to-patient variability, we took the example of vascular leak syndrome. So this syndrome is a side effect of immunotherapy with IL-2, and it normally affects 1/4 of the patients that are treated with it. So VLS, or vascular leak syndrome, is normally characterized by pulmonary edema and therefore a decreased barrier function and also cytotoxicity in the epithelium. So what we did is we took alveolar epithelial cells from different patients, we co-cultured them with endothelial cells on our chip, and then we treated them with IL-2 to see what happened. It was very interesting to see that only a subset of these donors were responding to the treatment, that actually only certain cells were having this decrease in the barrier function and also increase in cytotoxicity, as you can see here in the graphs. However, what we call non-responder or non-sensitive cells preserve both the tear function and cytotoxicity levels close to the controls. Nevertheless, what we could detect was that the secretion of cytokines happened in all the patients. So we could say that for sure both were responding to IL-2, but only those more sensitive would be more susceptible to have vascular leak syndrome. So these results show that our system can replicate this patient-to-patient variability, showing that it could also be applied in personalized medicine. As another example, we characterized our cells for the expression of a particular type of HLA, which is basically the histocompatibility complex, or MHC, in humans. And this molecule is responsible for the self and non-self recognition via the immune system. In this case, we were looking for HLA-A2, and we just took our primary cells and then screened them by flow cytometry for it, and we can see to the left some negative cells and to the right some positive ones, and these ones we would select and then grow on the chip. Why do we do this? So our interest in the HLA is because there is some antibody-based immunotherapy that requires particularly HLA types to work, and HLA are specific to humans, so this actually makes in vitro systems the perfect approach. So in this study, we wanted to study the effect of a T cell bispecific antibody, which brings together T cells and epithelial cells. Here you can see how it works. In this particular case, this TCB was supposed to be used for treating a certain type of cancer, and actually it had to be withdrawn in the late preclinical development due to a very high toxicity in primates. So what did we do? So we took the TCB and then we treated our cells with it. And what we could see is that we could actually replicate this toxicity issue that was found out later, showing a very strong barrier disruption. Here you can see the decrease in tear is very dramatic, and also increased secretion of pro-inflammatory cytokines. Actually, if we took them together, these results show that the lung chip technology can be of very big advantage in safety studies. So to summarize, I hope I have given you a flavor of how MPS are bringing increasing value to in vitro research, and this can lead actually to more predictive new approach methods that have basically two main benefits. First, they represent a 3D alternative in so that they can help replace animal tests. And besides, they gather human relevant data for more efficient and cost-effective molecule development. Besides, using NPS, we can target questions that are specific to certain species and are targeted, for instance, for humans. And this has happened, for instance, with COVID-19, in which the receptor for the virus could not be found in some of the conventional animal models that are used for research. Another example is the targeted immunotherapy I just showed before. In addition, NPS are also allowing for personalized or precision medicine, given that they can capture patient-to-patient variability. And they are also suited to studying minorities that are normally excluded from the drug development pipeline, such as those people suffering from rare diseases or certain other populations. From a technical point of view, NPS allow for conventional and advanced in vitro readouts that have been also developed for other in vitro systems, and this is an advantage that we can basically use the knowledge that we have from before and just integrate it into these novel in vitro systems. But of course, NPS still have a long journey in front because they are quite complex and they are also still not standardized. So one of the factors that needs to be taken into consideration is that we need to work on characterizing deeply how the cells are behaving in this NPS in these environments and how similar they are to the human body. Another important factor is their multiplexing capacity and how we can implement the different readouts. So this can be used in larger screenings, for instance, for drugs or for toxicants. And finally, currently there is a lot of work putting into establishing different strategies to standardize and validate the models. So hopefully NPS can be included as a standard procedure for industry and healthcare applications not so long in time. Thanks so much for listening. I hope you have enjoyed the webinar, and please just get in touch if you have any questions.